This lecture on extracellular RNA carriers in plasma has been composed by Tom Driedonks, University Medical Center Utrecht, the Netherlands, and Esther Nolte at Hoen, Utrecht University, the Netherlands. In this lecture, we will explain how to determine in which extracellular RNA carrier your RNA of interest is found. We will illustrate this by showing data of our studies to identify the extracellular RNA carrier to which Y-RNA in plasma is associated. We have already discussed Y-RNA in our previous lecture, but we'll give you a quick refresher why this is such an interesting RNA molecule. We know that Y-RNA is abundantly found in EVs from various cell types and biofluids. And Y-RNA is one of the most abundant RNA molecules in plasma. Furthermore, Y-RNA has been proposed as biomarker for cancer, cardiovascular disease, and inflammation. So to further explore the functional and biomarker-related properties of Y-RNA, it is important to determine to which RNA carrier the Y-RNA is associated. This image gives an overview of the different particle types that are found in plasma. So besides the extracellular vesicles, plasma contains protein aggregates and various different types of lipoproteins. For example, chylomicrons, they are the largest type of lipoprotein particle. The other lipoprotein particles are named after their buoyant density. VLDL is very low density lipoprotein particle. LDL is the low density lipoprotein particle. And HDL is the high density lipoprotein particle. With our studies, we tend to focus on EVs, but please be aware that lipoprotein particles and proteins may also carry extracellular RNA. So why is it important to define the type of exRNA carrier to which biomarkers of interest are associated? When you know what carrier contains your biomarker RNA, you can do enrichment for that specific RNA carrier to increase the sensitivity of detection and improve the diagnostic potential of your biomarker. Furthermore, you could hypothesize that the RNA in EVs may be a better reflection of the RNA content of diseased tissues compared to, for example, lipoprotein particle-associated RNA or total plasma RNA. So it's well worth investigating where your biomarker is located. In this lecture, we will discuss the complexity of exRNA carriers in plasma. We will discuss size and density-based methods for separation of exRNA carriers. And we'll go over RNase resistance testing to find out whether an RNA is inside or outside of extracellular vesicles. Finally, we'll answer the question of which exRNA carrier contains the full-length Y-RNase. We have shown before that there are many studies that detected extracellular Y-RNA in a wide range of biological samples. It is also clear that there is a wide variety of protocols that are used to enrich for extracellular Y-RNA. You can see that in this table. Most of the studies used ultracentrifugation-based methods, which enrich both EVs and ribonuclear protein particles. Some of the biofluid studies isolated RNA from the total biofluids. So while these methods will give you RNA to work with, they may not tell you what extracellular RNA carrier the RNA of interest is associated to. So back to our scheme, which depicts the different extracellular RNA carriers. You may have noticed that the extracellular RNA carriers in plasma here have overlapping sizes and buoyant densities. We can use size-based methods to separate large and small particles. We can use density-based methods to separate low-density lipoprotein particles and high-density protein complexes away from intermediate-density EVs. But only by combining size and density-based methods, a relatively pure population of EVs can be isolated from this complex fluid. Other fractions that are obtained throughout this procedure will be enriched for ribonuclear protein particles or lipoprotein particles. So let's discuss these methods in a bit more detail. Size exclusion chromatography is used to separate small and large particles based on size. Size exclusion columns contain a resin in which the large particles, here in red, elute more quickly than the small particles, here in green. When this method is used to fractionate plasma, the early fractions will contain EVs and large lipoprotein particles, whereas the later fractions will contain small particles such as LDL and HDL, protein complexes, and soluble proteins. It is already known for quite some time that these fractions differ in RNA content. In the paper presented here, for example, they determined the microRNA content of size fractionated plasma and demonstrated that specific microRNAs elute in early EV fractions, while other microRNAs elute in later protein fractions. Density gradient ultracentrifugation. That's a method that is used to separate exRNA carriers based on density. Here we show an example in which 100,000 G pelleted material from culture medium is separated further using a gradient. What we can see then is that EVs float to their buoyant density, whereas protein complexes stay at the bottom of the gradient. The bioanalyzer traces on the right show that both EVs and ribonucleoprotein fractions contain RNA, each of which have a distinct size profile.
So now back to our original question. Which extracellular RNA carriers in plasma contain Y-RNA? In order to answer this question, we used a combination of size and density-based separation methods. We started out with platelet-free plasma, which was obtained by blood collection in a citrate tube. We subjected this to size exclusion chromatography, during which we separated large particles, including EVs, in the early fractions and small particles in the late fractions. We then applied regular ultracentrifugation to separate the light lipoprotein particles from denser EVs and ribonucleoprotein protein complexes in each of the fractions. Finally, we applied a density gradient to the pellets to further purify the EVs. In our paper, we showed data on all of the 6 different fractions, but to make our case more understandable, we will focus on the EVs versus the small lipoprotein fractions. On the right side, we show a Western blot indicating that the EV fraction is indeed enriched for EV markers CD63, CD81, and CD9, whereas the lipoprotein markers ApoA1 and ApoB100 are present in the lipoprotein fraction. Next, we quantified Y-RNAs in these fractions, as well as 2 well-known microRNAs for controls. MicroRNA 16 is known to be present both in EVs and lipoprotein particles, and microRNA 122 is predominantly found in lipoproteins. This is indeed what we observed in our qPCR data. MicroRNA 16 was enriched both in EVs and LPPs, whereas microRNA 122 was only detected in lipoprotein fractions. So how about the Y-RNAs? In contrast to these microRNAs, Y-RNA1 was predominantly detected in EV fraction and not in the small lipoprotein particle fraction. This was also the case for the other Y-RNA subtypes. These were most prominently detected in EVs compared to the small lipoprotein particle fraction. You can also see that Y5 was relatively low in abundance, which corresponds to most of the literature. Next, we performed RNase resistance testing to determine whether Y-RNA was on the inside or on the outside of EVs. We subjected each sample to RNase plus protease or RNase protease and detergent treatment. If an RNA is located on the inside of an EV, it should be able to withstand RNase plus protease treatment. In contrast, the protein complexes or lipoprotein particles will be degraded by the protease, and subsequently the RNase will degrade the RNA. Only after adding detergent, the EV enclosed RNA will be accessible to RNase and degraded.
So let's have a look at the data.
What you can appreciate is that microRNA 16 in EVs is resistant to RNase and protease treatment, whereas the same RNA and lipoprotein particles was degraded by RNase and protease without presence of the detergent. Also, microRNA 122 in the lipoprotein particles was degraded under the same conditions. In contrast, EV-associated Y1 was resistant to RNase plus protease treatment. Thus, it is likely that Y1 is on the inside of the EV membrane. And in the paper, we show similar results for the other Y-RNAs. To summarize, we have shown that plasma is a complex biofluid that contains multiple extracellular RNA carriers. We have also shown that a combination of size and density-based methods applied sequentially are needed to obtain a pure EV population plasma. In addition to these techniques, RNase and protease resistance assays can be used to assess whether an RNA is exposed or inside of EVs.
To answer our research question, Y-RNA is present in EV fractions in plasma and is located within the lumen of the EV.
Thank you for your attention.